In a relaxed
Actually, I think we will listen to this for one time.
muscle, actin and myosin myofilaments lie side by side, and the H zone. In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
So just a touch on contraction. What's that?
There's always something wrong with this cookie.
Well, it's not my clip.
Well, the cookie needs to bring your cookie.
I think what it is people leave it on. The batteries get worn down very quickly.
If you eat your own cookie, then you'd actually take care of it.
Oh, this is good.
Well, that happened last time too. Sometimes it gets stuck on the animation.
Well, we got the lab that's due Thursday.
What about it?
Well, I can't open the file.
And if we have it due Thursday, we'll open the file in class.
I'm coming to the early lab on Thursday, and that's one of the 2 days I'm going to Mary's lab.
Talk to me later.
Okay.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. the head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
So this goes circular, but remember, you had to use ATP to get your head to cock and the movement, but also you use ATP to get the calcium pumped back up. So you're going to use more ATP than just what you see here. So you need the ATP for the muscle to contract, and you also need more of it for it to relax. Because the reason it relaxes is that the calcium volume concentration has gone down. Sarcoplasmic reticulum is like the endoplasmic reticulum, except in muscle cells. Okay.
And action potential.
Okay, contractions of skeletal muscle. These are done in vivo, and we're applying electrical stimulation to it. Somewhere in Mary's repertoire is a video of me with a neurologist from Hollister, Larry Grace, coming in and sticking needles in my arm, one up here, one down here, and running an electrical current between them. Kind of like acupuncture, but I mean, they're so fine that they really didn't hurt badly. But he was measuring the electrical, the nerve impulses moving down the arm, as he would look for, say, a carpal tunnel type disease. But again, here they're attaching little probes into the muscle and exciting them with electrical stimulation. So what you do is when you hit it once, you get the twitch. If you continue to stimulate, the twitches go together, you get what they call a summation. As you see, they build until the twitch is when a muscle quickly relaxes, contracts and relaxes, increasing the voltage, increases the strength. When a second shock is applied immediately after the first, the second twitch will partially piggyback on the first. This, you keep applying the voltages, and it's summation to a point where you get up to tetanus, where the muscle doesn't have time to relax. Now, there's two different types of tetanus. You have the tetanus that you get from bacteria, and you have the tetanus you get from something like this. The fact that in tetanus, in bacterial tetanus, the muscles still are not allowed to relax. And if you see a picture of somebody that has it, your heart back's terrible way to go. And if you Middle Ages into the 1800s, early 1900s, they didn't have a vaccine. You got a cut, you picked up the bacteria. There was nothing they could do about it. So, here, 5 shocks per second, 10 shocks per second, 60 shocks per second, you're up to a complete tetanus. The muscles aren't having time to relax at all. Finally, you're going to hit a fatigue where no matter how much you shock it, the muscles aren't going to do it. Asynchronous activity of the motor units. The muscle, all the muscles innervated by one nerve is a motor unit. If they all fired at one time, you wouldn't have any smooth kind of movement. So they're asynchronous. You have some firing at 1 millisecond, and a few milliseconds later another's firing, so that you can produce a smooth movement of that muscle. Trip. As the voltage increases, the number of muscle fibers used in vitro increases. When it reaches a maximum, you're at trip. As you bring in more summation, more muscle fibers, it'll staircase up until you reach a plateau. Forced velocity curve. The less weight you have on a muscle, the faster it's going to move. That's all this is. If I'm lifting this and I'm going like this, I can do it very quickly. If I've got a 25-pound weight in my arm, it can be a lot slower. That's force velocity. Force velocity. So if you look at the left, the 0 is the amount of weight. Load opposing contraction. The higher the weight, the slower the velocity of the muscle contraction. Isotonic and isometric contractions. Isotonic, muscle fibers shorten, tension is produced. It's greater than the load. If I have an isotonic contraction, I can lift something. I can make it move. If I have an isometric contraction, let's say this is a bag of cement. I try to lift it, my muscles are trying to contract, they can't because the force, the weight of that bag of cement is more than my muscles can move. You get an isometric contraction. They're trying to contract, but they're not shortening. Concentric and eccentric. A concentric contraction is like an isotonic. The muscle fiber shortens when the force is greater than the load. An eccentric contraction is like the isometric. Muscle may actually, an eccentric contraction is the muscle may actually lengthen despite the contraction. This is the type of contraction you would use if you were setting a very heavy object down gently. Your muscles are still contracting, but they're actually lengthening, and that allows you to lower a weight. Series elastic component, the non-contractile parts of the muscle, the tendons, are pulled tight when the muscles contract. The tendons, they have to take up the slack. The tendons are pulled tight before the muscle, before you can lift something. They're elastic, they resist this tension, they resist being stretched, and then they snap back to resting length. They absorb tension as the muscles contract. Now, a very important part of that is in your respiration, in your breathing. As you take a deep breath, your muscles are pulling the diaphragm up, creating a vacuum that sucks air into the lungs. There's no muscles that drop the diaphragm. You can't push it down. It's the tendons. It's the elasticity of the tendons that pull it back. Muscle strength determined by the number of fibers, the number of motor units, the frequency, and the thickness of each fiber. As you exercise, and your fibers become thicker and stronger, especially your alpha fibers, motor neuron, not alpha motor neuron, the alpha fiber, the fusel, the extrinsic, and we'll get to that. Length tension relationship. Tension is maximal when the sarcomeres are at their normal resting length. When they showed you the picture of a muscle, the Z bands are at a normal length, a few nanometers apart. When that is where it belongs, you have your maximum strength. Because if you stretch the muscle out, the amount of myelin that is overlapping the amount of actin for the myosin heads to grab is not as much. So you lose strength when the muscle is stretched. There are fewer interactions between the myosin and the actin. At a certain point, you don't have any. And you can't contract that muscle, not when it's stretched that far. Decreasing the sarcomere length, again, decreases the muscle tension, and you lose strength at that point. So there it is. You're looking about somewhere between around 2.25 microns to have the best strength in a muscle. You stretch it out, you lose strength. If you let it contract too much, you lose strength. Energy requirements in skeletal muscles. Rest and mild exercise from fatty acids. You guys go to the gym, you look on the elliptical trainer or whatever, and it has cardio, it has strength, and it has fat burn. Depending on how much you're using your muscles, you use a different type of energy. At rest or for mild exercise, you're getting a lot of it from fatty acids, break down to triglycerides. When you look on your exercise machine, your fat burn rate is not nearly as high as, say, the cardiac rate, the cardio rate, which sometimes makes sense, sometimes doesn't. For moderate exercise, you're getting more glycogen storage. It's taking glyconeogenesis, breaking down some fats, some lipids, some protein. Heavy exercise, you're getting it more from blood glucose. Intensity of duration increases your GLUT4 channels. Remember what insulin does? It allows GLUT4 to come out, the channels to come out of the cytoplasm and move to the plasma membrane, which allows glucose to flow into the cell. That's what insulin does. And that's how the GLUT4 channels work. More of them are inserted from the cytoplasm into the sarcolemma, which is the what? Plasma membrane of the muscle. And that allows more glucose into the cells. So here it's, looking at where it's getting high energy, medium energy, low energy, whether it's coming from glycogen, triglycerides, fatty acids, or glucose. Maximal oxygen uptake. You finish running a race, you're still breathing hard. You've used up the oxygen that's bound to the hemoglobin, you've used up the oxygen that's bound to the myoglobin, and your body is still oxygen deprived. So you stop running the race, your muscles aren't working, but you're replacing that oxygen. That's why you're still breathing. Determines whether called aerobic capacity or the VO2 max, and it varies greatly from people that are in shape or out of shape, couch potatoes or runners or marathon runners. And genetics has a big deal to do with this too. Acclimatization too. You're around here, your hematocrit, average is probably around 42 to 45. You go to somebody that's living at 13,000 feet in the Andes, their hematocrit probably runs 55 to 60. You acclimatize, the body's saying, I'm low on oxygen, I'm going to make more red blood cells, to carry out, to capture whatever oxygen I can. VO2, determined by person's age, sex, size, and athletic training. It goes from 12 to 84, and somebody that's in very good shape, that 84 could be 120 to 140. It could almost double. Lactic threshold. Remember, when you don't have the oxygen, to accept those electrons, and in the mitochondria, in the Krebs cycle, you don't have the oxygen there, what happens? It goes into an anaerobic, and it adds the hydrogen back to the pyruvate to make lactic acid. So that's what's accepting the hydrogen ions. Now, granted, this happens a heck of a lot quicker than in the Krebs cycle. This type of cycle happens a lot faster. But at some point, you're going to build up the lactic acid, and that's your lactate threshold. Anaerobic threshold. This is when at the point where your rise in blood lactate or lactic acid occurs. Now, your muscles operate pretty much anaerobically and when you start exercising for the first minute or two, until the body can open up vessels and start getting oxygen into the muscle. But that's not what we're talking about here. Oxygen debt. This is why you're still breathing when you get back, when you get done. Exercise oxygen is withdrawn from the reserves, and your breathing continues until you repay the debt. That's why you breathe so fast, even after you stop the running. It takes you a while to catch your breath. You're getting this myoglobin and hemoglobin reoxygenated. Phosphocreatine. We know that we're using ATP for muscle contraction. At some point, you start running out of ATP. The body only stores so much ATP. So it has a system with phosphocreatine, so it pulls the phosphorus off the creatine, combines it with an ADP to make more ATP. And this happens very quickly. Creatine is produced by the liver, kidneys. It stores, it replenishes, but it gives you that extra boost of energy when you're running. That extra ATP that you're going to need. ADP is kind of combined with phosphorus from the phosphocreatine. You're left with creatine, produced by the liver and kidneys, or contained in the diet. Generally, it's liver and kidneys. Phosphocreatine stores are replenished at rest. When it's replenished at rest, it starts taking ATP, making more ADP, and replenishing your phosphocreatine. In the same way, you keep breathing to replenish the oxygen that's being used up. So here, at rest, you can see the phosphocreatine, or the creatine is going, if you go from the creatine on the bottom, go to the left, ATP is made into ADP, and phosphocreatine is created, and it's stored that way. When in exercise, the phosphocreatine is dephosphorylated, and which turns ADP into ATP, and you've got the ATP to use as energy for the muscles. You know, as I said, that's one of the reasons I wanted to show you that little animation. You use ATP to contract a muscle, you use ATP to relax a muscle. Contraction is part of the power stroke. Relaxation is used by the calcium pumps to pump the calcium back out of the muscle and allowing the muscle to relax. Slow and fast twitch fibers. You have two different types, actually you have three different types of flat fibers. Slow is a slow contraction speed can sustain the contraction for long periods without fatigue. Rich capillaries got good blood supply, more mitochondria to produce ATP, more respiratory enzymes, more myoglobin. It's something for a muscle that's used constantly, and it's a muscle you don't want to have fatigue. And a lot of times you have no clue that these muscles are being used until something happens. Say, Laura, you're sitting at your desk and it's a really long, boring lecture. Of course, that's not my money, but that's okay. You start nodding off, what happens? Your head drops. Now, does anybody walk around saying, boy, I'm consciously going to hold my head up all day? These are your slow twitch type muscles, postural type muscles. They're being used constantly all your waking hours. And they don't, they don't necessarily fatigue. High oxidative capacity, called slow oxidative fibers. Myoglobin, they've got plenty of oxygen. They're called red because of the myoglobin. That's what gives the muscle its color, is the myoglobin, found in postural muscles. Fast type, faster contraction feed, faster fatigue, faster, fewer capillaries, less myoglobin, less mitochondria. They're called the white fibers. They have more glycogen stores, called fast glycolytic fibers, found in the stronger muscles. But you get your strength from them, but they fatigue easier. Your neck muscle is not that big a deal, unless you see an NFL football player who's used to wearing them. What is how much? 7, 8, 9 pounds, something like that.
Those bodybuilders who do
Yeah. Then you have the intermediate. Fast twitch, but with a high oxidative capacity. These all, the ratio of these fibers change with the amount of exercise. If you have a spinal cord injury, you'll lose the slow twitch fibers because you're not, it's a use it or lose it.
That's why they can't move their legs, or, I mean, I know that there's a lot of things where if they do that, that's why they can't like really sit up on their own, but they can still some of them can make conscious short movements.
The postural muscles are degenerating.
But that's why I think a lot of people think that if they're paralyzed, they just kind of like stuck all the time. They really have like seizures and twitches and stuff like that.
To a certain extent, depending on where it's at. And then, you see somebody that, is a paraplegic or quadriplegic, they're in physical training constantly, just to keep some sort of muscle tone. Because you also look at the muscle tone, and we'll look at it more when we get into the next chapter, or chapter 14, because the muscles are around, and we'll talk about it at smooth muscle at the end of this, but they're around your capillaries, and they're around your veins. And when you walk, there isn't enough pressure from the heart to push blood back up to the heart. It's contraction in the muscles around the veins, and a peristaltic type action, and valves, literally valves in the veins, that push the blood back up to the heart. You look at somebody, a traffic cop, somebody that's standing all day in one spot, not walking. Their legs swell. You need to keep moving. You need the muscles to massage in the veins.
Isn't that when people with stem attention, military, and pass out?
That's when they lock their knees.
Well, that also happens simply because the blood, and especially on a hot day, I've certainly seen it in some of the things that I've attended. You're on a hot day, your veins, your capillaries are expanded, your blood volume's low, and then you're standing there not pushing the blood back up to the heart. If your knees aren't locked, then you're using the muscles. So that slow and fast twitch fibers, you can see how they change with training, spinal injury. Your slow type, the ones that don't fatigue, extreme endurance, world-class marathoner, middle distance runner, they're much higher. The average person, but then look at somebody with a spinal cord injury. There's, they've lost most of that. Muscle fatigue is the reduced ability to generate force, the reduced ability to lift anything. 1, you accumulate extracellular potassium, reducing the membrane potential. You deplete your glycogen. You have reduced sarcoplasmic reticulum calcium release. everything here is just kind of getting, you're fatigued because the stuff's using up. The resources have been used up. You get an accumulation of lactic acid. And it says lower pH. They're not really sure that the lower pH makes that much difference, but it certainly is the accumulation of lactic acid. Increased concentration of phosphorus due to the phosphocreatine breakdown. At some point you start, the phosphocreatine is broken down, the phosphorus is just out in the cytoplasm, it's not attached to the ATP, not attached to the ADP, and not attached to the ATP, you get ADP. You don't have the phosphorus attached so that you have the muscle, the energy for the muscle. And the upper motor neurons, the neurons out of the cerebral cortex, the cerebellum, the pyramidal tracts, they've just been used too much, and they're they adapt a little bit, and you get fatigue. Adaptation to endurance, exercise training. You increase the ability to use fatty acids. Now remember, one chain of fatty acid, 16 carbon fatty acid, what does that give you? About 128 ATP? Because it's broken down. Each 2 carbon gets into makes pyruvic acid, and then it goes through the Krebs cycle, and then the electron transport cycle. So you get 16 carbon, you get about 128 ATP out of that. The more you can use that, one fatty acid molecule, you got all this energy right there. You have more mitochondria in cells in the muscles that are being exercised, so you have the ability to use those. Decrease in the non-oxidative fibers, the type 2, so you can use, you've got more oxygen stores. Increase in the number of mitochondria. The mitochondria is where your ATP is produced. How do I know? Oh, you weren't looking that up?
Oh, no.
Oh, I just thought you were checking. Increase in the number of mitochondria. The mitochondria is your powerhouse of the cell. That's where your ATP is produced. That's where your electron transport system is. And the more mitochondria, the more ATP. The more ATP, the more energy you've got for the muscle. Effects of endurance training improve the ability to obtain ATP from oxidative phosphorylation as opposed to an anaerobic. This is oxidative. Increase the size and number of mitochondria. You've got more muscles, energy generators. Less lactic acid. Increased myoglobin. Myoglobin stores oxygen. Increased triglyceride. Triglyceride breaks into your fatty acids. Fatty acids give you a huge amount of ATP per molecule. Increase lipase that allows you to use the fatty acids. More energy derived from the fats, less from carbohydrates. Lower rate of glycogen depletion. Improved efficiency in extracting oxygen. You've got the myoglobin, decrease in the number of fast fibers, increased in the number of oxidative. You've got more oxygen, you're not getting into lactic acidosis, your muscles are not being fatigued by it. Adaptation to strength training, hypertrophy. What's hypertrophy versus hyperplasia? Size. Hypertrophy, the muscles get bigger, the fibers get thicker. The thicker the fibers, more nuclei, stronger they become. They can split into 2 fibers. Increase in size requires titin, nebulin, and obscurin. I'm not going to go into it much more than that, but those are the 3 large proteins that are used in building muscle. Muscle repair, you have in skeletal muscles. You have stem cells called satellite cells located near the muscle fibers. They fuse together to repair muscles. Myostatin is a paracrine, which is a hormone that is secreted into the blood. It's secreted and acts on a tissue in the same organ, as opposed to an autocrine versus an endocrine versus an exocrine. Paracrine. Myostatin inhibits the satellite cells. We don't really know why. Animals that they manage to shut the myostatin off create a lot more muscle. Muscles decline with age. Usually the type 2 can be helped with strength training. Reduction of capillary blood supplies, endurance training, fewer satellite cells, increased myostatin. You don't have the satellite cells to repair, and you got the increased myostatin. I don't know why. I mean, that's where the research is right now. Yeah, but who wants to age?
Nobody wants to do it, but it's like what you have to do.
And it happens. Yeah, I don't argue that. If you, well, yeah, but look at it this way. If we can do research on this, and you can get muscle regeneration, you look at somebody, and at some point we can do it for smooth or cardiac muscle, then you have the opportunity to repair a heart. You have the opportunity to deal with the muscular dystrophy. You have the opportunity to repair somebody who's had damage and muscles that have degenerated. Now, 1930, if I walked up to Rebecca and said, "You've got pernicious anemia," lack of vitamin B12. In 1930, I would be giving you this. Literally, I would be giving you this. If I walked up there and said, you have multiple myeloma, you've got cancer, you're going to die. There's nothing they could do about it. Somewhere around 1935, they developed the B12 injections. And pernicious anemia is a disease of the past. There's things we do. There's reasons.
What about growth hormone for building muscle mass?
Well, they try that, but the downside, you get growth hormone is just like a hyperpituitaryism, and you get the acromegaly, the bone, the gigantism, and all that, and so it just doesn't work.
It makes everything grow.
Yeah, it can't.
It's not a proportional growth, and you can't target just muscle. That's all.
Yeah.
Yeah, okay. Now she's switched sides here. Okay, let's go on. Neural control of skeletal muscles. Lower motor neurons. Neurons that are coming out of the ventral horns of the spinal cord. Sensory feedback from muscles and tendons. Stretch, generally, is a stretch receptor. Stimulation or inhibition from the higher motor neurons from the brain. You get stimulation or inhibition. Golgi tendon organs. They're on the tendons. They respond, to the tension that a muscle puts on a tendon. Muscle spindle apparatus. You have your large, thick fibers, and then you've got thin fibers, intrafusal fibers. They form spindles. They run parallel to your muscle. The fibers, the thin fibers in the spindles really just kind of tell your muscle where it's at. And they're muscle fibers, and they're not big enough to move the muscle or the bone. They just keep muscle tone or muscle tension and help you to more finely coordinate the movement of the muscle. More muscles that have more control have more spindles. Now, remember, when we talk about the innervation of muscles, the motor plates, an eye muscle, a finger muscle might have 7 muscles per nerve. Whereas a muscle in the back or a muscle in the back of the leg might have 2,000 muscles innervated by the same nerve, because you don't need precise movement. The spindles work in the same way. The more spindles, the more control you have over the muscle tension, and the easier it is for you to do fine, delicate movements of that muscle. Stretching a muscle causes the spindles to stretch, and they give the muscle a more of a perception of where it's at. Tells us what's going on. The muscles contain thin muscles called intrafusal fibers. You have two types. You have the nuclear bag fibers and nuclear chain fibers. The nuclear bag fibers, the nucleus of the cell is kind of around the fiber. The nuclear chain fibers, the nucleus is kind of stretched out along the fibers. Then you have the primary and secondary neurons that wrap around the fibers also. So up here, you have, if you look at this, I am going to Radio Shack on this Tuesday and find a laser. Actually, I think I confiscated one from my son a few years ago. Probably have one. The big fibers up there. See the real thick ones in the upper right corner? Extrafusal fibers. These are the fibers that give your muscle strength. These are the ones that do the work. You go in and you have intrafusal fibers, which are the smaller, the thin ones. And then you can see the nuclear bag fiber and the connective sheath. The nuclear bag fiber, that's part of the spindle. And you can see how some of them are scattered up and some are wrapped around. And these are as the spindle is stretched, these pick up the stretching motion, send it up to the pyramidal tract, up to the brain, and it allows you to control and know where that muscle is at. Lower motor neurons. You have alpha, which innervate the extrafusal fibers. Remember again, the extrafusal fibers are the big, heavy fibers. They're the weightlifting fibers. The intrafusal fibers are innervated by the gamma motor neurons. Contraction of an interfusal fiber doesn't do anything. They're too small, they're too few, and they're too weak. It just provides muscle tone and tells you where, position and just how much stretch is in the extrafusal fibers. When a nerve goes down through the motor plate, it tells a muscle to contract. It'll do both alpha and gamma motor neurons at the same time, and that's called coactivation. Reflexes. We looked at that last Thursday. Skeletal muscles are usually voluntary, controlled by the higher brain regions. They can contract unconsciously in response to certain stimuli. That stimuli might be pain, that stimuli might be pressure, or generally stretch. When you took that rubber mallet and wrapped just below the knee, you stretched that tendon, which went back to the spinal cord, crossed over from a sensory receptor to a neuromotor receptor, and you got your knee jerk. And they use this in the book. If you step on a tack, you don't sit there and think, oh, geez, I got a tack in my foot. Maybe I ought to lift my foot. You step on a tack, and this is bisensory. You step on a tack, one leg is going to come up, the other leg is going to extend so that you can maintain your balance. And this is a reflex. You don't even think about Simplest. Sensory neurons and then synapsing on a motor neuron in the spinal cord, your knee jerk. One synapse, monosynaptic. Helps maintain the optimal resting length, because what happens, you've got your knee bent, it's stretched, you hit the knee jerk and you stretch it even more, it comes out, so it isn't stretched anymore. Muscles are not stretched. You're striking the patellar ligament, knee jerk reflex. One synapse, monosynaptic. Stretch on a muscle stretches the spindle fibers. This activates the sensory neuron, either your primary or secondary, sometimes both. Activates the alpha motor neuron. Motor neuron stimulates the extrafusal muscle to contract, and the stretch on the spindle is reduced. Now, you just artificially stretched that spindle by hitting it with a hammer. That's why you got the knee jerk reaction. Not the pain. If you do it right, it doesn't hurt that. Knee jerk. Striking the patellar ligament. Spindle is stretched, activating a sensory neuron. Sensory neuron activates an alpha motor neuron in the spinal cord. Alpha motor neuron goes back, stimulates extrafusal, the weightlifting muscles, and boom, you've got your leg moving, you've got your knee jerk. Golgi tendon organs, they're on the tension, they monitor the tension of the tendons. Stimulates interneurons in the spinal cord, interneurons stimulate a motor neuron, tension in the tendon is reduced. Your knee jerk is a patellar tendon. By just tapping it, you're adding a little more tension. And boom, you've got your knee jerk. Disynaptic involves more than one synapse. This is your Golgi tendon. Tension on the tendon activates the sensory neuron. Tension again is tapping it with the rubber hammer. The sensory neuron stimulates an interneuron. Interneuron stimulates a motor neuron. Alpha motor neuron is interneuron inhibits a motor neuron, and the tension is again reduced. Hang on, let me look at this for a second. So what it's done is this at one point you're stretched, and the Golgi tendon is firing to say stop stretching, essentially. This is a disynaptic. It says sometimes if it's stretched too much, your muscle will actually, because of the Golgi tendon, will relax. Reciprocal innervation. In the knee jerk, interneurons are also stimulated in the spinal cord to inhibit the antagonistic muscles of that limb. If I've got to flex, something's got to try to tell the triceps to relax. This is your reciprocal innervation. If I consciously say I'm going to flex this, something unconsciously is going to tell the triceps to relax, or else your muscles are fighting each other. And there are some disease processes in which that happens. More complex reflexes require control of muscles on the contralateral limb. That's stepping on the tap. My right foot's on the tap, my right foot's going to lift up, my left foot's going to lock or activate more of the muscles so that you don't lose your balance and fall down on the floor. One, muscle stretch. This is reciprocal innervation. Muscle stretch, it activates the spindle apparatus. Antagonistic muscle contracts in a stretch reflex. Agonist muscle contracts, antagonistic muscle relaxes. You're going to do a curl, your biceps contracts, your triceps relax. Double reciprocal innervation, you see when you select, it says, this is from the book, you step on a tab. Flexor contracts and extensor relaxes to withdraw foot. On the other foot, contralateral side, the extensor contracts and the flexor relaxes to support the weight. This again is in your book. That's a double reciprocal innervation. Upper motor neuron control, precentral gyrus through the pyramidal tracts. The cerebellum receives information from the muscle spindles about position, postural type information. Golgi tendons, how much, what your muscles are doing, where they're at. It inhibits a region of the basal nuclei, the red nuclei, and the vestibular nuclei. The basal nuclei also act to inhibit motor activity through the rubrospinal tract, and we will get back to that later. Okay, cardiac and smooth muscles. Cardiac and smooth muscles are involuntary, regulated by the autonomic nervous system. Contraction is due to myosin active cross bridges and stimulated by calcium. Cardiac muscles are striated. Look at them under a microscope. They're striated. They have the actin and myosin filaments forming sarcomeres. Contraction, again, occurs by means of the sliding filaments, which is what we saw before. Unlike skeletal muscles, which are kind of long, your cardiac muscles are short, branched, and connected via gap junctions or intercalated discs. Now the thing about that is the gap junctions, if you remember, are an electrical junction between the cells. So when you fire one cardiac muscle cell, they're all going to fire. That's why if you have somebody that has, say in the SA node, the cardiac pacemaker isn't working right, you can put a pacemaker in, and you can put it in one spot in the heart, and you fire that pacemaker, and the whole heart's going to contract. As opposed to having on a skeletal muscle, like we looked at the summation and all that, you would have to put, without this type of setup, you would have to put probes all over the heart if it was a skeletal muscle. With a cardiac muscle, you fire one, they all fire. The fibers are short and branched, connected via gap junctions, which they're called intercalated discs because when you stain them, they look like a disc at the end of the muscle. What they are is an electrical gap junction, connecting the muscles electrically, and like I said, you fire one, they all fire. There's your gap junctions. And here they're calling it the intercalated disc, but they're the same thing. Okay, the myocardium is a mass of cardiac muscles connected via gap junctions. Action occurs in any cell, can stimulate all the cells in the myocardium, behaves as a single functional unit. The atria compose one myocardium, the ventricles compose another. Now, we'll get into this in chapter 13, but the atria, the valves open, the atria drain into the ventricles. The ventricles, the atria collect blood from either the lungs or the body. Put it into the ventricles either pump it out to the lungs or out through the body. If they both contracted at the same time, nothing's going nowhere. So what happens is your stimulation starts at the SA node, sinoatrial node. It spreads across the atrium, and the contraction of the atrium pushes the last 20 or 30% of the blood out of the atrium into the ventricle. Then from the ventricle, when the ventricle contracts a millisecond later, it pushes the blood out through either the pulmonary artery to the lungs, or out through the aorta down to the body. But if you look, and we'll look at this further, the electrical current comes out of the SA node, and then it hits the AV node, atrioventricular node, which slows it down by a millisecond, a couple of milliseconds, a few milliseconds, so that the ventricles contract just a little bit after the atrium. So they have time to fill, then they contract, and then the whole cycle starts over. Pacemaker. Cardiac muscles can produce action potentials without innervation. Begin in a region called the pacemaker, the SA node. Now the problem with this, without autonomic control, is, yeah, you lose a heartbeat, your heart will go on beating 20 to 30 beats a minute. In other words, unless you're sleeping on oxygen, you're going to be in trouble. So that's why, you see people coming in like that, you're going to put a pacemaker in. Heart rate is influenced by the autonomic nervous system and hormones. What hormone kicks your heart into high gear? Epinephrine comes out of where? Oh, come on, he's not. Yes, he's asked that already. He's adrenal medulla. Yeah. Fight or flight state. Unlike skeletal muscle, the voltage-gated calcium channels are not directly connected to the calcium channels in the SR, sarcoplasmic reticulum. Remember, in skeletal muscles, we have the tubules that connect them. Instead, the calcium acts, instead of going down through the tubules and into the muscles and allowing the attaching to the troponin and allowing the cocking and the muscle movement to take place, in the heart, the calcium comes in and it activates a second calcium gate that releases about 10 times more calcium, which gets in and causes the depolarization. So instead of calcium, unlike skeletal muscles, the voltage-gated calcium channels are not directly connected to the calcium channels in the sarcoplasmic reticulum. The channels act as a second messenger to open the sarcoplasmic reticulum channels, and that's the same as endoplasmic reticulum in the other cells, non-muscle cells. And this is called a calcium-induced calcium release method. Excitation, contraction, coupling is slow. Your heart doesn't react as fast as other muscles. Okay, calcium in cardiac muscle. You have the calcium released, then you have the calcium gate channel, which is allowing calcium in, and once inside, it reopens up some other calcium release channels, which allows the troponin, binds to the troponin and allows the sarcomere to contract. Smooth muscle. And we're going to get back in chapter 13 and 14 is circulatory system and heart. We will certainly be coming back to that. Smooth muscles found in the vessel walls, bronchioles, digestive organs, urinary, reproductive tracts. It's autonomic. You can't control it. Produces peristaltic ways to propel contents of these organs. The smooth muscle not only goes the length of the organ, but it will also wrap around the organ. So that's how you can produce a peristalsis. That's how you can put pressure on the bladder to urinate. That's how you can, or how it reacts to pressure on the bladder to urinate. It's how it moves, food through the stomach. Also, it's how in fight or flight or cold or warm, the muscles, the smooth muscles, autonomically can shut off blood to various areas of the body. In the fight or flight syndrome, the smooth muscle shuts off blood to the surface. It shuts off blood to the kidneys. You want that blood going to the muscle, going to the liver, getting glucose out, going to the brain, going to the heart to oxygenate the heart. This is all done by smooth muscle. I mean, you have 5 to 6 kinds of blood, and it's like you were talking, Rudy, earlier. Hot day, your capillaries are dilated. You don't have enough blood in your body to fill all the capillaries at one time. If that happens for some reason, you lose blood to the brain and you faint. So all the time in your body, these smooth muscles are contracting, closing off capillaries. You ever seen somebody scared to go white as a sheet? Yeah? Your fight or flight syndrome, your smooth muscles have cut off all the capillaries to the skin because you're getting ready to run. You're scared so bad. And the blood's going to the muscles, it's going to the heart. This is all what the smooth muscles are doing. No sarcomeres. They attach to the cell bodies. They have some myosin, some actin. The thing about smooth muscles is with the myosin filaments, you can stretch them and they can still contract. With the skeletal muscles, like we talked about earlier, if you stretch it so that actin and the myosin are not overlapped, there's nothing for the myosin heads to grab and contract the muscle. Smooth muscles don't do that. I mean, you can get your bladder, extended 3 or 4 times, the muscles, and uterus during pregnancy, 8 to 10 times, and it will still contract. And I'm sure you'll find out soon. Smooth muscle. Instead of the filaments running the length of the muscle, your filaments some are running the length, some are wrapping around. Thick filaments, thin filaments, cell bodies. This allows not only a movement down, but a squeezing or peristalsis to move things down the vein, the intestine, wherever. Excitation, contraction, coupling begins with a rise in intercellular calcium concentration, some of it from the sarcoplasmic reticulum. Now, crosses the membrane, voltage-gated calcium ions are open, but the calcium in smooth muscles, there's no troponin. In your skeletal muscles, the calcium comes in, it binds the troponin, moves it out of the way. ATP combined, you get your power stroke. Smooth muscle doesn't have any troponin. So the calcium comes in and it binds to something they call calmodulin. It's essentially the same thing in the smooth muscles. And then that activates a myosin light chain kinase, which phosphorylates myosin light chains, a kinase, again, phosphorylates. And this allows the myosin to form its cross bridges and have a power stroke. The thing about smooth muscle as opposed to skeletal muscle, once the skeletal muscle starts going, that muscle fiber is contracting. With this calmodulin and the myosin light MLCK, myosin light chain kinase, you can get a graded reaction. in a smooth muscle. You can't get that in a skeletal muscle. The reason you have different activity of skeletal muscles is the summation of more muscle fibers. Smooth muscle you can actually gradate depending on the amount of calcium that's in there. So here you've got the membrane potential. You've got graded depolarizations till you hit a voltage gated channel, and then you get until you get it opened up and you get an action potential. But then you get the calmodulin and the myosin light chain kinase and the phosphate, and then you get your cross bridges, phosphorylation of the myosin light chain kinase. It phosphorylates the myosin, you get your cross bridges, and you get your contraction. If you have less calcium, you have less contraction. Again, it's a graded reaction. Calcium is pumped out using calcium ATP as active transport pumps. Again, you need ATP for the muscle to contract. You need ATP for it to relax. Then the calmodulin disassociates from the MLCK. Phosphates are stripped by myosin phosphatase, and the muscle relax. Single unit. Multiple gap junctions that make neighboring cells behave as a unit. What did we just talk about in the single unit? What organ would have a single unit? Heart. When one fires, they all fire. They display pacemaker activity. By stretch or autonomic innervation. The heart is stretch. The heart is autonomic innervation. Only a few cells in a single unit receive acetylcholine stimulation. And that would be in the pacemaker area, like of the heart. Muscarinic acetylcholine receptors respond by closing potassium channels. Multi-unit require individual nerve innervation. There's no pacemaker activity. Go-gam gap junctions. Erector pili muscles, skin, ciliary muscles in the eye are multi-unit. They each, if you look at that diagram, the erector pili pretty much had its own nerve innervation, as did the ciliary muscles in the eye. Autonomic innervation. Neurotransmitter is released along the autonomic neuron from varicosities. I think that was a question on your test. You get a number of smooth muscle cells are stimulated at once. Synapse en passant, autonomic, fight or flight syndrome. When one goes, boy, they all go. In your sympathetic system. And we've seen this before. We saw that in when we first started talking about the muscle, the nerves. There's your very eye. You see the bulbs along, running up along the muscles. So when one fires, they all release the neurotransmitters, and you get that stimulation en passant. Okay, I think you guys really want to start circulatory system tonight?
No.
Okay. Are you working at?
Are you at Gavilan in Santa Clara?
No. You got the midship?
Mm-hmm.
That's one thing nice about maybe not in
Yeah, you don't have to do
the I
that.
have worked on midship for 10 years. Of course, I have run most of the instruments in the lab in 10 years. They wouldn't want me on that. Okay. If you want to stick around,